Our next speaker is the wonderful Grant Smith, who comes from Te Rangahaumārangai, Climate Food Research, although he lives on South Island, very fine in the north. Over to you, Grant.
Thank you.
Almost. I need my mouse to be working. Hold on a second. What do you need?
Presentation mode.
Yeah, I'm getting there.
Yeah, so I'm going to talk a little bit about what I consider to be an wide host range obligate pathogen. Yeah, okay. Well, this is not can't read it that close. So it's a multi-host eukaryotic obligate pathogen, which in theory doesn't exist. That pandemic strain is a very large host range. There's also the South African strain, which also has a large but not as extensive host range. And you know, when you look at it, they appear to be quite considerable exceptions to Flor's hypothesis. Gene-to-gene hypothesis in terms of co-evolution of a complicated pathogen like rust and its host. So Peter Dodds, who's worked on rust in Canberra Sorrow for decades, and his colleagues. Peter published this work a few years ago, Flor's Enduring Legacy. I've actually seen Flor's desk and chair. They're still in place at North Coast State University. That's Peter's, well, before it became a university, but he did all his work up in North Dakota on the flax rust, so linen rust, and published a whole lot of outstanding papers describing the genetic relationship between this pathogen and its host. And you'll notice those dates starts in 1935 and proceeds through to 71. Next slide is, this is before the discovery of the molecule DNA and how hereditary and genes worked. So most of this work was done without full actually understanding any of the molecular biology or having any concept of how those genes were actually working, but he could describe the genetic system accurately. And his hypothesis is the gene-to-gene interaction between pathogen and the host. And that's that remarkable photo from nature that led to the discovery of the double helix from Franklin and Gosling. So Peter Dodds, a couple of years ago, gave a keynote lecture at the ABS meeting in Adelaide. And Peter's worked for a long time on rust of agricultural importance. And I spent some time talking to him saying, how does this rust operate? And he said, I do not know. We do not know how this rust actually has such a massive host range. In a non-compatible reaction, the rust will be stopped. Won't be able to stop and interact with living tissues. So, so this is drawn from the back to the front. Basically, we started over here. We're just trying to understand a little bit around the biology, and ended up over here through numerous grant applications and various sources and lots of contributions from lots of people. And part of it initially was to describe, was there resistance in the New Zealand species, the mutaceous species, and and what is the basis of that resistance? And can we understand molecular biology of disease of this really unusual? As far as I'm concerned, it's an enigma. It doesn't fit anywhere in our understanding of rust pathogens. So there is resistance in naive hosts, i.e. hosts that have never ever seen this pathogen before, they have not fully evolved. And we actually have reasonable amounts of resistance in, for example, in this case, in our Manuka populations in New Zealand. What we did find, importantly, was that there's actually two types of resistance in Manuka. And the initial work that we did suggest that they were not on the same chromosome. Those lineages were not, sorry, those resistances were not linked. One was a stem resistant to infection, to a stem infection, the other one was to leaf. And that map there shows that through the provenances that we screened Manuka from, there were different levels of resistance in those provenances. So some of the more resistant ones were some of the ones on the east coast, of the North Island. There was no, yeah. And in part of that, we also found that there was effectively at that time almost no resistance in Manuka. We found one possibly resistant plant at that point in time. Not good news. So I'm a classically trained plant pathologist. Anyone that was at APS who recognises that? Can you repeat the name? Oh. What? See how much attention being paid to me? Anyway. That's RKS Wood, Imperial College in London. He's generally considered to be the father of physiological plant pathology, i.e. how do pathogens cause disease. He's also my grandfather in terms of my descent, my lineage. He's my supervisor's supervisor. So that's why I have an interest in how pathogens cause disease, and it flows from this chap here. And we normally describe interactions between hosts and pathogens with a disease triangle. We need the right conditions, a pathogen and a host, but it's in the right conditions that we'll get expression of disease. Okay, so disease is the expression of system. So over 2 COVID lockdowns and various other misadventures, we, with our Australian collaborators, were able to do some initial experiments that I wanted to do to try and understand gene expression of both the pathogen and the host, particularly in really early on in the infection. This pathogen gets up and running pretty quickly. And what we're able to show that in terms of resistance, that the resistance of the leptospermins that we had in our experimental setup, and they had already been phenotyped, we already knew what their resistance phenotype was. There was a strong family effect, so there was some different genetics playing out between the this is an non-co-evolved system, so you expect to see different genotypes. There was also a change in gene expression over time, especially in the ones that were actually interacting with the pathogen. And the phenotypes linked back to the descriptions we had generated around the saying what genes they did not contain. So, what's that? Right. So I got particularly interested in effectors, and I think others have talked about effectors. They're relatively small proteins, they're cysteine rich, and their job is to get in there and let the pathogen gain access to the resources of the plant. So they alter the plant host physiology for pathogen benefit. And I think I'm talking about the second genome there, Furio, I think.
Yeah, it would be.
Yeah. Let's update that slide. But it was phased, so I had thought, and there were over 1,200 predicted candidate effector genes in that genome assembly. So there was a lot of opportunity to go and explore. And you can see that effectors in particular run as a series, in a stylized model, run as a series of pulses. So different effectors do different tasks during the infection process and manipulate the host. You've seen this photo before, Rebecca's presented it before, but I just want to focus. These are our, I've drawn these bars around our favourite effectors. This one, 1260. Down here, you'll see it's got some strong red. So red is high in expression, and then the next time period, so this is over 24 hours, it goes blue. So which means it goes up and down really quickly. That really, and the one below it does something very, very similar in 948. Now, Jonathan's going to tell us a little bit more about 1260 as a protein tomorrow, because we are interested in this one for quite some time. And then 5292 also is another one which is showing that sort of pattern of hot and cold very, very quickly over the initial time frame of infection. So this is mapped out, 1260 mapped out. You can see it's also its expression level, as you'd expect. L1 is resistant, L2 is immune resistant, hypersensitive resistant, susceptible. So it shows higher levels of expression in the susceptible host plant because it's gained control of the mechanism. It hasn't been kept out. So we've always been interested in this particular one. And part of this work involved trying to get an infection process working, and colleagues at the University of Canterbury had developed a synthetic replica leaf surface. Now, rust don't normally like to form hysteria on surfaces when you put them on a glass slide, they just won't, they'll just germinate and do nothing. On this polydimethylsiloxane, there's an opportunity to test whether we could actually use that as a synthetic plant surface to see if we could actually get the rust to believe that it was infecting a plant tissue. And the answer is yes. So these are some of the photos that were taken in Brisbane. They're quite beautiful. And remember the first telephone conversation with Helmut Taggart over these. He was absolutely excited beyond belief. The thing that says IP up there is infection peg. It's actually thinks it's on a leaf and it's actually trying to drive an infection peg into that leaf. So they're a great experimental tool for investigating rust physiology and pathology. And they're described in Beck's first paper, figure 3. So really interesting and exciting. So yes, and that's described in that paper there with the initial work was done around targeting 3 housekeeping genes with double-stranded RNA. A little bit more work. We were really constrained about some of the work that we are constrained about working the way we want to work on introduced pathogens in New Zealand. So they become deregulated. So again, work that was done in Australia. This is 5292, another one of those vectors I noted before. Again, if you target it with double-stranded RNA, you basically don't get infection on the leaf. There was one bit of noise and one of the effects. And again, 10948 in 1260 in this one. When they were targeted, they seemed to it seemed to reduce the amount of germination of the spores. So it does raise the question, we think of effectors as being key for pathology of the system, but they also might be important for the biology of the rust in terms of spore germination. So we possibly don't quite understand completely what the effector is at this point in time. So going back to 4. Beck and the crew in Brisbane did a lot more work, and in 1260 it's always been our favourite, and basically did some more work with 1260 and found that it was actually belongs to a gene family. It's not actually a single, and it probably in the latest, it's probably spun out a few variants, I think, but we didn't have that in the second genome assembly. And it's a gene family of intergenomic variants in a tandem repeats flanked by transposable elements. And more importantly, it may be the driver of the host range, this pathogen. In terms of the intergenomic variability in the family, it facilitates the host range versatility. So at least we've got some ideas now and some experimental proof as to how this pathogen may be able to operate as a multi-host range pathogen and basically make Flor's hypothesis null in this particular case. And I'll leave you with a couple more of Pal's magnificent photographs of the pathogen. Thank you.